Hi, everyone. So as you're coming in, we've got a poll launched. We're just interested to hear who's joining us today. So it's asking career stage and what continent you're coming from. And just to review very briefly, so we had a lot of people joining us. We're very excited about that. Stefano and I didn't know what to expect when we decided to run this workshop, and we had a lot of people, so we have to do this muting just to make sure it's everybody can listen and participate, can hear. And so we're going to ask for, we can take comments in the comment box during the talks. Otherwise, in between the sessions and at the end, we'll use the raise hand feature and try to go through people's questions in the order they come up. Okay, I guess I'll get started. I can end the poll and show you the results. There's a couple of people. There's a little acceleration. Okay, so share these results. So yeah, so it looks like it's kind of evenly split between graduate, postdoc, and then postdoc. And some, predominantly folks coming from North America. Some of you woke up early in Australia. Maybe that'll change as the session goes on. Okay, I'm going to switch over. So I have a couple introductory slides. Okay, and I just want to introduce. So today I'll give some introductory slides. I'm Mike Love. I'm an associate professor of genetics and biostatistics at UNC Chapel Hill. Stefano, do you want to introduce yourself?
Yeah, I'm a postdoc at the Walter and Eliza Hall Institute in Melbourne, Australia, under Tony Pappenfuss Laboratory.
Thank you. And so, Stefano and I have interacted a lot in the bioconductor ecosystem. So we met each other probably on Slack, I guess, and started communicating. We both have an interest in tidy analysis for genomic data. And so we've been kind of kicking this idea around of having a workshop together, and December worked out. So I'm going to give an introduction, just a very quick opening about why tidy analysis might be useful for genomic data, and then I'm going to talk about a package called plyranges that was developed by Stuart Lee, Michael Lawrence, and Dai Cook from Melbourne, from Australia at least, and then I'm going to show some work that we've done at UNC with a package called nullranges. So let me go to these introductory slides first. Okay. Looks good, Stefano?
Yes.
Okay. So, this is not the right way to do it because it auto plays. So data organization depends on what you're trying to do. And so this might be a perfectly valid way to represent data if you're trying to visually indicate these outliers with the yellow highlighting, while I think a lot of us who are more focused on programming and working, programmatically interacting with data would maybe get a little cortisol in our blood when we see this type of presentation of data, because we know it's going to be kind of a pain to operate on this. We have to extract out the color and all this kind of stuff, and it's not ready to operate on. And actually, while I'm going through this introduction, I'm going to launch a second poll. It'll be going in the background, which is, I have to stop there. Okay, second poll. It's kind of about familiarity anyway, so it goes along with what I'm talking about. Okay, so instead, many of us, I'm sure you're familiar because you came to this workshop, are interested in rather keeping the data in this tidy format. So in this case, we have one row per observation, one column per variable. We operate on the data sequentially through a series of pipes. And so I thought this was like a nice quote that, unfortunately I can't see the comments. Oh, okay. These slides are posted, but I'll share it in a little bit. Okay. Pipes rank along the hierarchical file system, and regular expressions as one of the most powerful yet elegant features of Unix-like operating systems. And so in R, we use this pipe command instead of this vertical bar, but essentially it's the same idea. So we have the following verb-based operations that we are probably many of us familiar with from dplyr: mutate, adding new variables; select, picking variables; filter, picking cases, so rows; slice, picking cases based on position; summarize, to reduce to summary statistic, you could say; arrange, changing the ordering, and group by, performing operations on groups. And so a useful paradigm that I'll be using a couple times today is grouping data and then summarizing. So for example, with that data before, filtering out outliers, grouping by these combinations, and creating a mean value. So that's what we get. And we can, especially useful for exploratory data analysis, is to pipe directly into plots. So create a new value, and then directly like plot it and faceting. So genomic range data is often already tidy. And then I guess like to set up Stefano's session is like, it's going to be an interesting challenge to work with matrix shaped data. But range data is kind of already tidy. Sorry if you're getting some noise in the hallway. I don't know if it's bad. Okay.
No,
okay. And so, what are the advantages here? So in terms of, from the perspective of bringing people into genomic data science, and so like teaching or mentoring, many people who are coming into genomics are already familiar with dplyr and ggplot2. So you're not teaching this from scratch, but actually the verbs are already familiar. I think it helps make code more readable, so you can avoid like having to be creative with intermediate variable names. And so the code is not necessarily shorter, but the aim is for it to be more readable, like human readable. And I think these are kind of deeper reasons, but motivations behind thinking about writing tidy code for genomics, is that it encourages exploration. And in genomic data, it's very large, high dimensionality, with cells, like high number of cells as well. And so you really should be encouraging exploration as much as possible, that there's not going to be like a one-stop shop for like extracting the results, but we should be exploring the data as much as possible. And I think it encourages that. It's possible that it can encourage efficient coding. So to the extent that you can do one pass, like one function call, you end up with fewer calls out of R to C. That's maybe a more subtle point, but it's possible that it encourages more efficient coding. It might be more code to do something simple, but then that simple code tends to generalize well to more complex cases. And I would argue, from the point of view of like developing software, that it's actually easier to maintain if you allow users to do their own thing in the middle of a workflow, you can make your software very extensible without having to like think ahead and plan all the potential things that someone might want to do. Just very briefly, I'll have a little bit of like, this is now getting into this package called plyranges, which is working with genomic range data with dplyr-like syntax. This package developed by Stuart Lee. And so you can like enter into our ecosystem very easily. Like there are these dedicated functions with like read underscore, or you can, use this like as ranges function, which will take like a data frame like object. If you have your ranges written out with like colon and dash, you can use the separate function and then create ranges. So it's very easy to get data into our bioconductor. And then once you've got it, this plyranges package allows you to use these verbs that you're familiar with. So like take the top 3 and pick a column, like a metadata column here, like the signal, for example. And then, kind of more interesting things you can do. So like, so we have these peaks. These were like H3K, K4 monomethylation, I believe, from ENCODE. Now, for a set of query regions, these 1 megabase tiles on chromosome 1, say we want to find overlaps and perform some kind of computation based on the overlaps. So that operation is considered a join in plyranges. So we're basically joining these two sources of information by match. It's just instead of a match of like an ID column, it's now a match based on a genomic overlap. So you can think about how would you pick, for example, a top scoring peak per tile? So what verbs would you use? So I said we might want to do a join based on overlap. So one way to do this, so we can like select a column of interest. This is not necessary, not strictly necessary, but just makes the output easier to look at. We perform the join. That brings in the metadata columns that we had on these tiles, and it annotates the peaks with the overlapping tile ID. We can then group by tile ID, so that then our peaks are kind of grouped by what tile they overlapped, and we could just slice the max score. So, to the degree that you're familiar with the dplyr verbs, you're just doing the same thing you would normally do, but this is just happens to operate on range data. So, some operations are even simplified more. So if you want to like count the number of peaks that fall in each of these tiles, you can just add a column that counts the overlaps, where the dot will specify the self within this command, and you get that overlap count as a new column. So I'm going to pause there. Let me end the poll and share the results. And so let's see. So there's a mix of very couple people have never used a tidy language. So that was probably a really quick, too fast presentation for you. But luckily, like 91% of you have at least seen tidy before. A number of you are not familiar with the bioconductor classes. Looks like 20%. So that's okay. I will try to slow down now with the bioconductor classes, but luckily a number of you are a little bit familiar at least. And then it looks like a third have seen nest and map before, which was a question Stefano was interested in seeing.
Michael, you have just a couple of questions in the chat that I'm not able to answer because.
Oh, sure. Yeah, I'll how cross-compatible are the plyranges functions? Yeah, unfortunately they conflict. And so if I'm working with plyranges and dplyr, I kind of like I load the one that I want to use more often last. You can always use a prefix like that if you to explicitly call one. But this is a kind of an unfortunate situation right now that they do conflict. Maybe we can come up with some kind of solution right now, but we don't have one. So yeah, and the overlap, the functions that have overlap are the plyranges functions. Actually, all of these, because they're operating on granges objects, are plyranges functions. Okay, I'm gonna stop sharing the poll, and then so this was a very quick introduction to plyranges and how you can kind of do the familiar things that you know before, but operating on range objects. So like here, I just showed you counting overlaps, and on the previous slide, I showed like picking the top scoring peak in a set of tile regions. So now I want to switch over to enrichment analysis, which was the title of our talk, the title of this talk. And let me see, so I'm sharing screen. Hopefully this works. Okay, great. And actually now I have the comments, so that'll be useful. Okay. So I want to talk about tidy enrichment analysis, which is figuring out if like sets of features in the genome are near each other, or especially near each other. And the idea behind this, the idea of this package came out of a workflow that I worked on with Stuart Lee and Michael Lawrence called Fluent Genomics. We were working on doing an enrichment analysis, and we thought, well, it would be nice if there's a way to come up with null regions to do comparisons, to ask if the enrichment is more than you would expect by chance. And so that idea spawned this package nullranges, and we purposefully didn't want to make this crammed into plyranges, but have a separate package that really just does one thing, which is to create kind of background or control regions. And then at UNC, two graduate students stepped up and were interested in working on this. So Wentz and Mu in my lab here, and Eric Davis in Doug Fanstill's lab were interested in kind of developing the software. We also had a lot of input from people in the bioconductor community. So Mikhail, Stuart, obviously, and Tim and others on this Slack channel were interested and gave us input and looked over the code and such. So we've got two preprints that go into more depth about what I'll show today. It was, oops. Okay. And also I want to highlight that a lot of the diagrams I'm going to show right now are using another bioconductor package called Plot Gardener, which was also developed by a student in the Fanstill lab, which is Nicole Kramer. Oops, every time I click on the comment box. For working with large single cell data sets, do you see people making use of Parquet? I'll leave the single cell for Stefano. So I'm going to primarily just talk about ranges for the next 15 minutes or so. Okay, so here's a setup of a genomic analysis. Let's consider a set of features. These are called original features. For example, they might be like ataxic peaks, and they have a score associated with them. So I'm coloring them based on some metadata that's attached to each feature here. And I might be interested in asking if these features are closer than expected to genes, and maybe like are higher or lower scores accumulating near these genes. And I'll just say, because I simulated the data, that the answer is no. They're just randomly distributed, uniformly, I should say. So they're not actually distributed with respect to the genes at all, because I simulated these features that you see on the screen. So I simulated actually a wider set of features than I'm showing. So I'm kind of zoomed in to a patch of chromosome 4, and there's a wider range on chromosome 4 where I'm creating some features. So one way to ask if those features are appearing near genes, or if there's this particular distribution of scores near genes, is to create some background to compare against. And so one way we might do that is to just shuffle the start position of the features in the region, maybe excluding some regions like repeat regions or something, or maybe centromeres or telomeres. So many have written papers about how uniform shuffling is not a good background model. And so there's a number of papers in this null ranges package vignette that go into depth on like why that's not a very good background model. So maybe you could think to yourself, like what considerations are not taken into account by the shuffled features compared to the features I'm showing. So I would argue one is the clustering. So they tend to occur near each other and they kind of accumulate, and there's regions where there's less of them. And another is that the way I've simulated these, if they are near each other, they also have like a similar score value. So they have some correlated metadata, and they're also clustering in the genome. And so we might want our background or our control set to take that into account, have those properties. And one way that's been proposed to do that, to have a background set that contains those types of properties, is to sample blocks of data with replacement. So this also comes from the time series literature. So if you want to create like null or background time series, you can sample blocks of time. There's a paper from Peter Bickel and others going back to 2010 about this idea of using the block bootstrap to sample blocks of data for genomic analysis. And so the idea is you just sample with replacement from the original data set and create a new synthetic set of features. And so it's with replacement, meaning that you can resample blocks basically every time you grab them, you go and put the features back. So what's been preserved here, we've preserved both the clustery nature of the features as well as the correlated nature of the metadata, right? So if we had just uniformly shuffled, or if we kind of permuted the metadata, we would have destroyed that structure of the data. So I'm going to do this operation now. Let me name the genes G. I'll name the original peaks P, and I'm going to show some code for operating on this with plyranges and nullranges. So the data I'm showing now is, well, the original data is simulated. The shuffled and bootstrap tracks that's an actual bootstrap, a block bootstrap of a larger region of data, which is why you've got there's some features you see on the top track that are not in the bottom track because there's a larger region here. So you can easily create these bootstrap sets with the boot ranges function, and it has these arguments. So you have to choose the size of the block, and we have some recommendations for that. You have to pick the number of iterations, and then I won't talk too much about this, but it's important in some analyses to choose a segmentation. So for example, here's an example of a paper of a genome segmentation, and that allows you to control for large scale feature density or things like that. So you can choose to only bootstrap across segments that are of the same state. So here, in this example down here, maybe you don't want to bootstrap, you want features from state 3 to stay in state 3 on other chromosomes, for example. So we can do all these kind of plyranges like statistics and do it for the original data and do it for the bootstrap data. So for example, let's say we want to count the sum of the scores for each gene. We're going to go and find the overlapping peaks and sum up the scores for those peaks. You can really be creative and come up with any kind of test statistic here. S1, and this gives you this data frame. The NAs are for the genes that don't have an overlap, because I did a left join rather than an inner join. An inner join, those would be not present in the output. So because this is a simulation, and I told you there's no association between those peaks and the genes, I can just draw more data from the original distribution. So I just do this 50 times, and then I concatenate that into a long set of ranges with the bind ranges function. And I add a column, which is going to be called iter, ITER, which tells me the iteration, like which simulation was it. So there's 50 here. That's done with this .id argument. And then I can do this analysis over all the iterations. So this is kind of getting back to my first point about instead of doing a for loop and having 50 calls to find overlaps, the idea here is to stack the data into a long format and have a single overlap call to keep track of that with a column, and then perform a group by operation to get your statistics at the end. And so here in this case, I'm actually grouping by the gene and the iteration. And then I've added an extra column called type, and that'll be just used later. So right now I'm not really using that argument. I'm just making a single violin, but I'll use that argument later. So this is the distribution over 50 iterations of what typical values are for the sum of the score when I go from a gene to the peaks that are overlapping it. And so at this case, I'm actually removing the non-overlapping cases. So there could be some like NA values or 0 values here that I'm not plotting. So I'm going to do that 50 times. I'm also going to do 50 times shuffling the one original data set. So I can resample the original data from the simulation distribution, and 50 times I'm going to, from the original data, shuffle, do a uniform shuffle, and do a block bootstrap. And so again, the paradigm is like stack it all into a long object with a column that tells you what the type is. So now there's 3 types. There's like the simulated data, and then the 2 types of ways of creating background sets. And then I have basically the same code as before, where I'm doing a join, I'm grouping by the gene symbol, which iteration, and the type of range, whether simulated, shuffled, or bootstrapped, and then I can pipe it directly into a plot. And so the point here is that if you were to just shuffle the features and consider that a good background model, you would definitely overestimate the significance of your data, right? Because I told you at the beginning, this is null data. There's actually no association. There's no association of these peaks with the genes. They're created regardless of the gene locations. So you see these large values, like values above 10 and below -10, and those are occurring under the null. But with the simple shuffling, you don't see those extreme values. So your null model would be kind of overly, you'd be too excited about those cases when you saw them. Whereas because the bootstrap preserves the distribution of the data and like the correlated metadata, you do see these more extreme values. So just to say, this is a really quick analysis. What's missing? I told you I was doing the inner join. So there's all the cases that there was no overlap, and it's possible to add those back in. Afterward, you can use the complete function to kind of add back in missing values that weren't there. There's a question about whether there'll be a recording, and yeah, we'll post a recording of this to YouTube. And there's a question about what the shuffling looks like, and that shuffling the genes. I see. Yeah. So that's always a question. Actually, Aaron Quinlan was answering this type of question just earlier today. I saw on Twitter. So like there's a kind of directionality to this. So you have to kind of pick one feature set as like the anchor of the analysis. Yeah, so you'll get actually different answers depending on which set you start with. So you kind of have to answer that question based on what hypothesis you want to ask. So just to review that, and then I'll talk very briefly about a second set of control regions. So the key mechanisms here is don't have blocks of code where you copy paste the same code, right? So we were able to do many different analyses in one chunk of code. We iterated over, like we had these bootstrap iterations, we had different types of overlaps like bootstrap or shuffling or original, and we were able to do it all with one long set of data. And so another way to do that, if you want to do the observed data and the bootstrap data, you can set like the iteration to 0 for the original data. That's another way to have it all in one go. It's a single join overlap, and then we usually have some kind of group by operation, and then a summarize, and then complete to fill out anything that was missing. So I mentioned there's another type of way of specifying control regions in this package. And so we kind of developed two separate branches of functionality. The one I've shown you so far was on the right side here, where and the kind of the key distinguishing features between these two ways of thinking about control regions is whether or not there's a large pool of features that we can draw from to create background. If there is, then we don't have to create these block bootstrap regions, but we can select from this larger pool. And so the idea there is there might be some confounding variation that describes the features, and we can use that the distribution of the confounding variables to pick good background set. So this set of work is done by Eric Davis and Doug Fansteel and others. And so just a quick introduction to the idea here. We have a large pool of ranges, say, with metadata. We might have multiple types of metadata here. There's like the size and the color of the circle. We want to find a small set that from the pool, from the background, that matches what we call a focal set. So focal set is like the peaks of interest in this peaks example. So I'm going to redo this plot that I had before. So there are some focal features. They have a certain like distribution of color and of proximity to the genes. And actually, in this case, they are very near start sites of genes. That's how I simulated this data. And so suppose we want to compare the focal set, some property of the focal set, to some background or control ranges, maybe internal properties, or maybe the overlap with genes, say. And so, but we want to control for something like maybe the distance of these peaks to genes, and maybe also the color, which is here denoted by the score. The score, which is denoted by the color. So if we have a big pool, a big set, we can model what makes a focal range special. So suppose we have a much larger pool to draw from. Here, they're all stacked up because I'm zooming into a very small region, but this works well when you're thinking about the whole genome. And so let's say we want to control for the score, which is the color, and also maybe the distance to the transcription start sites. So using plyranges, we can add the distance from the features to the TSS. That's really easy. We're just defining the TSS. That's two commands, anchor and mutate, and then adding the nearest distance adds a column called distance. And then in this case, I'm also taking the logarithm of the distance. So it's not as heavy right tail. Okay, so then we can build a model for what makes the focal set special. And you just use like a design formula. So you're saying that you think score helps you be in the focal set and the distance. And those are the things that we want to control for. There's a number of different methods here, but just to show you like what would that give you? It would give you a match set, which is drawn from the pool, but looks kind of more similar to the focal set. And so then it enables you to make these more precise comparisons between like say focal and genes and the match set and genes. So you can see that they tend to have this like similar scores, and they also tend to be near the start sites of the genes. Okay, Eric has implemented a number of methods. These are all described in the vignette. And we have a number of plots to make sure that we did an okay job with the matching. So you can see like the solid blue and the dashed blue line are near each other. And I want to stop there, just acknowledge again the work from graduate students, so Eric Davis and Wenson Mu, and all the folks who gave us a lot of feedback on Bioconductor. Okay, I'll stop there. Maybe if there's maybe a minute for questions, but then we can swap over to Stefano. Or if there's not, we can also just take questions at the end. Maybe you should just switch over, and if there are any questions in the chat, I'll take them as they come.
Okay. Great. Thanks. Was very interesting, first of all. Let me share my screen. A second. Here we go. Okay. All right. So I will go forward with tidy transcriptomic, the other face of the coin. So I will the material I show today, it's a rendered R package, basically, which is available online. I will pass the chat. And this is the rendered version, but you can install the package and run the code yourself on the actual GitHub repository. It's an R package. All righty. All right, so this material is more extensive, that what is more detailed that what I will explain, but of course, again, we can go back, you can go back to it and explore it further. And has been produced in collaboration with Maria Doyle. All right, so very quickly, this is about tidy transcriptomics and focused on single cells. So how you can analyze, manipulate and integrate transcriptomic data using this tidy principle that Michael explained. Briefly, what you will learn is how to use this ecosystem, which is made of several packages, including tidy Surat, tidy single cell experiment, and also tidy summarized experiment and tidy bulk for pseudo bulk or bulk analysis. And what you will not learn is the foundation of the actual single cell sequencing technology and the fundamentals of single cell data analysis and tidy data analysis. So I'm assuming you have a foundation of this. And I would like to stress that this workshop is not intended to teach you how to do the analysis, so to give you the best way how to do the analysis, but just how to, using these adapters to probably do more efficient analysis. There is, I have a couple of introductory slides. Michael did a great introduction of the tidy R philosophy, so I don't need to reiterate. I just show you that we have a couple of resources. So the blog about tidy transcriptomics, where you will find the manifesto, so the principle that we are following. And we have a couple of publications for some more formal material about tidy bulk for bulk and pseudo bulk analysis, and tidy surat, which is the interface for the surat data container to the tidy ecosystem. So about the rtidy, I will want just to show you what is the most used data type, which is the data frame called table that this tidyverse ecosystem made quite powerful, which is going toward being almost a database rather than a simple table. So you have this observation as rows and variable cell column that can have simple types such as characters, but also more complex types such as plots, ggplots. You can have other data frame inside, so you can start to see how you can do nested analysis, applying functionality for each of these rows. You can have linear models as columns, but also surat and single cell experiment objects. So you can see the power and flexibility of the table data structure. Just to mention the bit of the big picture of the tidy transcriptomic ecosystem, we have developed a part for single cell, which is at the bottom, and for bulk, which is at the top. So starting from bulk, we have this tidy summarize experiment that is an adapter to this popular container from Bioconductor to the tidyverse ecosystem. So not only now you can interact from your summarize experiment to the Bioconductor ecosystem, but also with the tidyverse, and that makes very powerful. And also we have our most extensive package is this analysis workflow that follows tidy principle, which is called tidy bulk. So you have all these connections here. Similar things for single cell. We have two adapters, which are tidy single cell experiment and tidy surat, that not only now they can interact and work with their respective frameworks, but also now you can use the tidyverse ecosystem on them. And today I will focus on these mostly single cell, but we will also see some pseudo bulk analysis. Very briefly, most of you might know might be familiar with this very too popular single cell data container, and this is how they look like when you display on the console. So you get a summary of the information included within. Of course, in the back end, they are very complex hierarchical objects. What about the analysis? Well, each container works with mostly with their respective ecosystem, which is the Bioconductor community, for example, Scrum and Skater packages, and the Surat ecosystem that also partially contributed by the community. For data manipulation, these two have their own grammar. So for manipulating, you have to learn some details about one or the other. And while for very simple manipulation, you might have quite simple grammar. When we start to do more complex manipulation, like happens often in real-world analysis, the code becomes a bit more convoluted and hard to understand. I will just make you an example for single-cell experiment. Let's suppose you have your data, which includes your counts, and your a metadata table, and you want to join this metadata table with your single-cell experiment and select the cells for which this information is present. Well, you have to use cbind, and you have to use match within to be careful to match correctly the two data sets. And you have to use a subset command to subset the cell that have a certain information, okay? So now, how this compares to the tidy paradigm. Now, if we load tidy single cell experiment and tidy surat, and we print the same object on screen that is untouched, we see a different representation. So we see data frame representation where the cell related information are exposed to us, including metadata and a reduced dimension. And you can notice that the two representations now look the same. So we are kind of harmonizing how these two data containers appear to us and how we can interact with them. And not only they appear as data frame, but they behave as data frame. So we can use all the tidyverse infrastructure to manipulate, filter, join, visualize, and so on. What about the analysis? Well, nothing changes here. So these two packages, for example, they don't carry analysis. So you can use, you can apply all the same analysis were used before now. The objects are completely untouched. And what about the manipulation? Well, you can see now that you can use elegantly the tidyverse functionalities. And you can do quite complex operation, as I gave you example before, of joining and subsetting in one go in this case, as with powerful commands such as inner join based on sample in this case. And you can also note that now the code you apply to single cell experiment works with Seurat objects as well. So the cost of switching to one framework to the other in case you want to apply specific tools is not that big. You don't have to necessarily learn specific grammar to manipulate your object. Just a quick list of the operations that are available. So we include most of dplyr and tidyr functionality, but also ggplot and plotly. So now you are, if you want to do custom visualization, you can pipe Seurat and single cell directly in ggplot and plotly. Just to specify what these packages are and what are not. So tidy adapters that I show are not data containers. So data containers remain the one you are using and are not analysis tools. What they are is a data interface that allow manipulation, integration, and visualization. So the question, can we go from tidy Seurat to Seurat, is not relevant here, as we never leave Seurat or single cell experiment. Okay, so hopefully this clarified a few initial things. Again, feel free to, between one session and the other, especially if you have any question, very welcome.
I guess, Stefano, you could launch the poll that asks them what they prefer. It would be kind of interesting to see who showed up.
Yeah. Can I, or
I think it's on the bottom of your, you know, if you want to. I think it's on the bottom of your, but maybe it's hard to see if you're sharing screen. Don't worry about it.
Let me see. I see familiar. I don't. I see just 3 polls here.
Okay, I'll try to do it just for fun.
All right. So there are 3 sections. Hopefully, I will be able to go through all of them. Otherwise, the material will be there. So this is for you to become familiar with this type of ecosystem. Now, let's introduce tidy Seurat, and all this code, as I mentioned, will work with tidy single cell experiment if you use that object as well. Here we go. Thanks for that. Well, I
sure, just for fun.
Okay. All right. So let's load our Seurat objects. Let's print it on screen. By default, we see the standard view with a few statistics, summary statistics about the object. Now let's load tidy Seurat and print the same object on screen. Well, now, as I explained before, we have all cell information exposed. Why we focused on cell-related information for single cell compared to bulk is because the cell is the very key element in single cell compared to bulk, where maybe the genes are the key elements. We see a few common, again, summary statistics, but again, all the information here. Now you can also use tidyverse.
Oh, sorry to interrupt.
Well, very balanced. Okay, that's good.
Yeah.
We've never done it. I see. So for who has never done single cell, take this as a resource. You will go back probably and explore further as well. Now we can use on this representation on our Surat object, the tidyverse ecosystem manipulation, in case you are very nostalgic about the original visualization, but you still want to use a tidyverse functionality. You can toggle the visualization back. In this case, we like the tidy representation better. This Surat object is untouched. You can use whatever was working before with Surat is working now. So as example, simple example, we'll apply assay, which is a Surat functionality to our object, and we get what we expect. I will just scroll through a few commands you can, from dplyr and tidyr you can use. Let's start with filtering our object. Let's say we want cells that are in G1 phase, as simple as that, and you can see that we have less cells here. Let's say we want to subset some of the metadata columns for some reason, for displaying them, exploration, or just making the object smaller. We can select our column that we see here, and that's our result. Our reduced dimensions are read-only column, so they cannot be modified for obvious reasons, and are always there for you to filter on, visualize, and so on, do analysis on. Just to show you that when we do selects and any commands that modify the object, we get back a Surat objects that has been updated internally. So if we select some metadata columns and we explore the actual metadata, we see that the metadata is just 2 columns. Okay, so it's been updated for us. We can create and modify columns. Let's say here, very simply, we are creating a lowercase phase column. As such, we are showing that for you, and you can see here the 2 columns. And again, the metadata will be updated for us. Now, a bit more more realistic example. Often when we import and read single cell data, we might read it from a path that might include metadata information itself. So for example, sample ID. Now, thanks to quite powerful operators from tidyr, in this case, we can take our file column, use the extract from tidyr, create a sample column with an arbitrary regular expression. And we have, and here I show the results, we have extracted the sample pretty easily from our column. Again, I'm not creating temporary variables for it. So you can imagine for cleaning and pre-processing, you can have just one pipe and having an elegant code and not clog your system with temporary variable. In some case, they are very big if you have millions of cells. And other operators from tidyr, quite powerful, is unite, separate. So we can merge 2 columns or separate them. Let's say we want to create a unique sample identifier. And in this case, we are merging sample column with this alphanumeric column. And here it is. So on and so forth. You can imagine you have a big list of operators, okay? If anybody has any question, feel free. Otherwise, I will proceed with the case study. Well, I will answer the question that was asked before about these frameworks for big, large single cell dataset for Brian. So yes, I'm not familiar with all of them, but especially with the Human Cell Atlas, even other frameworks have been developed. So these kind of database and on disk object will become more and more popular. Some other is dbplyr, which works with databases, and another one is called TileDB that it will be pushed on from the Chan Zuckerberg initiative. Okay, so I notice instance filtering will both work on Surat object and dplyr by mistake. No, so these packages work quite well with the original dplyr and tidyr because they don't create other R generics, meaning that they don't conflict. Okay, there might be some edge case that, you know, as we are developing, we might pick up, but generally don't worry about that. If it's a table, select will work as if it was a table. If it's a tidy is a surat, will work as a surat. Okay, so the abstraction takes care for you. Cell types are from Symphony. No, well, I mean, I haven't shown any. You mean the data set? No, are not. I'm not sure what you mean specifically, but let me know. All right, so signature visualization. So you can read more details here, but in brief, I was doing an analysis of single cell from blood of breast cancer patients. And after the whole analysis, we were asked to focus on a subset of T cell, gamma delta, which are not so well characterized in this disease. And so we had the task of identify them, isolate them, and reanalyze them to characterize them. Okay, so that's the goal here. So we have our data set. Although by default, we show you and you can interact with cell related information, you can add to your data frame the features to do all sorts of calculations, visualization, and so on. So what we did to start with, to explore the literature, an example is in Pizzolato that provides a very small gene signature and provides the arithmetics of how to calculate these signatures. So these are 6 markers. And we use joint features to add these markers to our data set, as you can see here. And now we can do operation on these markers. We calculated signature score. So as you can see, we can have complex arithmetics in a very elegant way. No need to create, again, temporary variables here, because each of our gene is a column. So the simple formula in this case is a sum of positive markers that we rescale subtracted to the negative marker that we rescale. And here we are creating a new column, which is called signature column, which I'm showing you here. You can see here. Okay, so it's a double type. Now, well, what's the next thing we might want to do is to visualize these cells based on this signature. In this case, we are using, again, surat functionality. So we can pipe alternate between tidyverse and surat functionality with no problem. And you can see here that we might have a cluster that is likely to be gamma delta T cell. Okay. There are many visualization tools that are great, that are ad hoc from the community, surat and so on. But sometimes for publication purposes or for whatever reason, often we want to do custom visualization. And in this case, tidysurat makes it pretty easy to do so. So there is no a big cost in going custom. So we can pipe our object that we modified directly into ggplot. In this case, we are simply reproducing the same plot, but you can imagine the ggplot ecosystem is huge. So now everything is ready for you to use. In this case, because you map our 2 of our column, we can simply call them, color by our new signature score, set up aesthetics, and we can see here that the same cluster pops up. We can, of course, have all aesthetics for us as size, dimension, labels, and so on and so forth. Okay, so the next step might be, all right, we might have identified this cluster. We want to explore them quickly to see how to proceed to recluster and so on. And you can imagine you can try to gate these cells or filter by a threshold of your signature. And I will not go into details here for in interest of time. When we do longer workshop, I show you the interactive procedure, but there are other tools that allow you to, for example, create a gate column calling tidy gates. And in this case, you will be prompted to draw your gate, and you will have a new column in your data set that you can then filter on. So you can imagine you go draw your gate, press OK, and do whatever you want with that column. In this case, we are filtering where gate is equal 1. You can draw multiple gates and so on. Another quite trivial method that obviously is not optimal is to, for the purpose of this workshop, to go on. Let's say we want to filter based on a threshold, and this threshold is great. Just selected the cells we want. Okay, as very simple. So we keep piping our object to reanalysis. In this case, after we filter these cells with our favorite analysis pipeline, we can normalize, select variable feature, integrate, and so on. Okay, so as you noticed, you can do all this in just one go. So if any step doesn't satisfy, you just change steps. So no variables are created here. When you want, you save your variables. When you don't want, you don't need to. Just because tidyverse offers powerful operators that allow you to do that. In comparison, if we want to do it with base R, that's what we will need to do. So we create some count positive, applying our scaling, selection and rescaling, our count negative, selection and rescaling. We create our signature score, updating the metadata with our arithmetics, and we do our subsetting. Okay, so when you apply this procedure many times in your daily work, you will see the big difference. It is the same difference that you have from base R, from common analysis to tidyverse. So if you start, you won't go back. I almost guarantee. Okay, so I will go to the question. Last thing about this practical application. Not only we allow to pipe directly to ggplot, but this is valid also to plotly. So you can create interactive plotting. So let's say, this is a quite neat example. Let's say we usually visualize 2 UMAP dimension or t-SNE dimension. Let's say we want to get a different perspective, and sometimes it's very useful to visualize in 3D, so 3 dimension. We can calculate our 3 dimension. In this case, we run UMAP. All right, we have this object already ready when we do live coding. You can see that our object have been created, UMAP 1, 2, and 3. And because we have this column-based structure with metadata, reduced dimension in the same paradigm, you can pipe to plotly, specify our 3 dimension, specify our coloring. All these are columns. And when the data set is quite complex, I found it helpful more than once to have a feeling of the heterogeneity in this new dimensionality. And so we can hide cells and be a bit more interactive with that. Okay, this is just another nice example, as there is no cost into producing quite complex visualizations. There are some homeworks for you if you feel with energies after this workshop. I won't go through this now. All right, I think I'm good with time. Okay, there are a couple of questions. Is the pipe same? Okay, so I'm using here the base pipe that works with, I think, R 4.1 forward, and just because is a bit nicer to see, to print out. But for the purpose of this, doesn't make any difference. The Magritte pipe is a bit more flexible in a way, and the backend is a bit different. But if you Google the difference, there are very long posts that explain everything. But in short, no, there are no huge differences. Okay, again, I see there is a discussion. Great, you already take on that. Okay. Single cell experiment as cold data, raw data. I wonder if tidysurat can handle both in similar ways, such as filtering genes, certain genes versus certain barcodes. All right, so we have tidy Surratt that deals with Surratt and tidy single cell experiment that deals with single cell experiment. Yes, so call as default, we visualize for single cell related information, so mostly call data. This is different from bulk, as I will show you later. About the genes, where can you filter based on genes? I guess you can use single cell experiment functionality to filter on genes. Okay, so every comma that worked before works now. So I would open the brackets and do that, simply because this framework is not focused on genes. And for practical purposes, for single cell, I guarantee that you will not feel this lack of gene focus, because most of the operation is about calculating features about the cells, subsetting cells, analyzing cells. But then, yes, you can apply all the, if you want to do differential gene transcript abundance analysis, you apply the same procedure as before. There's been an attempt to conduct flow cytometry using, yes, I mean, single cell experiment, or tidy gate. Yes, you can do, the classical gating that flow cytometry is common with. And I guess, but I think there are also resources for flow cytometry in R, but for sure you can have flow cytometry data into these containers and apply the same thing we are doing now. All right, so pseudo-bulk analysis. So the last part of the presentation, Michael, and how much do we have?
I say you have as much as you want. I think this is really helpful, and then we can see how many questions there are. I'd say keep going.
Yeah, it will be 5-8 minutes, this one. All right, so I've explained you single cell. Now, they're actually the biggest platform that we created is Tidy Bulk, is a full analysis framework for bulk analysis, and in this case pseudo bulk, that follow tidy principle. So often I won't go into detail about too much the pseudo bulk. There are links here, but in short, often is both powerful and convenient to reduce the complexity of your of the single cell data to pseudo bulk. So we aggregate cells, for example, for a sample or a combination of a sample and cell type, summing their transcript output per gene, or averaging them. This depends on you. But this is very good to understand the biological and technical variability across samples, for example, or use some very established tool for hypothesis testing. For example, differential gene transcript abundance, if you want to test that. And I will show you an example here. All right, so Surratt, and there must be other packages that allow you to aggregate cells together to create pseudo bulk. In this case, I've coded this quite a long time ago, and we provided with our workshops. It's quite neat aggregate cell function as allow you to declare an arbitrary set of columns that you want to aggregate according to, and outputs a summarized experiment. So now you go from one container to the other. And this is also work for single cell experiments. So you go from single cell experiment to summarized experiment. As you notice, we have still the same tidy abstraction as before. This is because we are using tidy summarized experiment. So you change the object, but thanks to these tidy adapters, everything I said before works now. The visualization is quite similar. The only difference that for bulk data, genes are the very key element. And so we have a further abstraction in a way that we have a feature column, which are genes, a sample column, and our whatever metadata, which is sample related or gene related. So we have a long representation. And this is all, you know, we can use a lot of tools, everything that works with summarized experiment, tidy bulk, and this is quite neat to also to visualize. So ggplot works very well with this abstraction. All right, so as I mentioned, now tidy bulk is open for us, and we have a large number of functionality, and each of these wrappers underlie very established tools. And I mean, you can have a read, but you can do basically all standard operation, such as scaling, filtering, testing differential abundance, deconvolution, survival analysis, gene enrichment analysis, so on and so forth. So quite comprehensive list. Let's suppose we have our single cell data. We want to use pseudo bulk to estimate the association of gene transcription across conditions. Here is treatment for each cell type. Okay, so we have 10, 20 cell types, few samples. We want to compare the treated samples versus the untreated for each cell type. Okay, so each cell type might react in a different way to treatment. So that's what we want to test. Okay, so we have created our pseudo bulk data, okay, with this command. We have an our summarized experiment, and now we might want to group according to cell type and do an operation. Okay, usually we can you know, the philosophy, which is the standard philosophy, which is, you know, kind of base R related, is to split our data. We created a list and we apply something on that list. But the tidyverse ecosystem provides surely the most powerful paradigm for functional programming in R, as R is functional programming at its core. So if you are not familiar with nest and map, I suggest to explore those. You find information in Google, and to apply, because your analysis will change. Very, very powerful. I will show you just here. Now, it's might be complex to understand at the beginning, but trust me, just dive into that. So we have our summarized experiment. We can use, thanks to tidy summarized experiment, this nest, which is from tidyr. And here we are nesting based on cell type. What we get out is a data frame where one column is cell type, and the other column is our summarized experiment that now are grouped by cell type. Okay, and with this map function that I will show you, we can do operations on this column, you know, serially. Again, without creating temporary variable, in a very you know, functional way, self-contained. So is also very good to not create bugs about our environment and so on. I want to show you what's in this first object. So in this case, I take the first row, I pull this column, and this is a summarized experiment that I just show, just including one cell type in this case. Okay. Let's see, as I said, we want to use tidy bulk to do differential expression analysis. So we have our nested object. We use mutate to update our column. We could have created a new column. In this case, we are updating the same. And this map function that takes as input our column, it produces an output, which will be also our column, because we decided to update, and does some operation. Okay, in this case, X is our input. We filter based on abundant transcript. We test our differential abundance. This includes many methods and many functionalities. So if you read the documentation, you will be, it's easy to use and it's quite flexible. It can have contrast of any kind and so on. In this case, very simple. We are comparing binary treatment. And we create a scale abundance column for visualization later on. So a new column will be added to our data set. and we save, in this case, our object. How does it look? As before, except now these summarized experiments have been updated. Again, I want to show what the first element is a summarized experiment with now all statistics added to our object. Those are automatically saved in raw data and are exposed to us. So again, we can do anything with those: filter, do analysis, summarize statistics, and so on. And also you can see the RNA scales. Here you have the RNA column. Also RNA scale have been added. So now, for example, we can visualize and do other analysis. To conclude this very small example, let's say we want to select the top variable, the top, well, differentially expressed genes for each cell type. Okay, so we have our nested data set. Again, we go through with map, create a new column, which is called top gene, and simple select transcript information, arrange by p-value. We want the first one, and we get the feature. Then we want to do some visualization. So we want to draw a box plot just for the top gene. So we filter our object. So again, we have another block code that does another operation. And we update our object, taking our object plus our top gene, and we filter one with the other, applying map. Okay, so these, obviously, if you're not familiar, these .x, .y are kind of, very specific syntax. But again, have a look, and if you learn about this, it will become very natural. Again, I'm doing quite complex things here, not create any temporary variable. So everything is self-contained in each block. You will have much less bug as I found in my own analysis. What about these objects? Well, similar as before, we have our two columns plus with this top gene. And now this summarized experiment will have just one gene in them. And what we do at the end of this very small example, we want to plot this data. And so again, we use map to add a plot column, using our summarized experiment and our cell type to add the title. Summarized experiment again is .x, and our other column is .y. And we simply pipe, thanks to tidy summarized experiment, this object directly to ggplot. Okay, and so we, as common as quite standard, we put treatment in x-axis, our scaled abundance on the y, we choose the aesthetics, so on and so forth. And if we look at this table, now we have a plot column. So you can see that this is almost working as a database. We have many types grouped by whatever we want. And if we pull this plot column, we can see that for each cell type, we have the top gene here. Okay. This is a quite very trivial example, but you can imagine you can scale this up. This works for single cell, so you can nest single cell data, which is even more helpful probably than for bulk data to do cell type specific analysis and so on. Okay, so this is just applied to pseudobulk. I'm done. Again, the link is in the chat, so you can go back, install the package, run the code, and last thing, I'll just pass we have quite a few workshops now that are in this repository. Some of them are much longer than this, so welcome to have a look. Thanks a lot.
Let's open it up for questions. So I think people should be able to, I think, raise hands. And if you can do that, then Stefano could, I guess, unmute you. Or I could just allow, let's see. Or people could ask in the chat if that's not working. I'll start with a question. So at the end, you had the multiple plots, and then you could like, I guess, like stitch those together with like patchwork or something. Could you also pipe direct, like what if I want a facet on the cell type instead of like having multiple ggplots?
Yes, you can. So, well, in this, there is a technicality in this, because when you can unnest the whole data set, and when you unnest, all the summarized experiment will be stitched together again. So the samples have been must be unique, but you can do, while summarized experiment, of course, puts constraint on the validity of the object. For visualization purposes, you can convert, inside your map to a simple table. So your summarized experiment will look the same, but will actually be a table. You join everything together and you facet based on whatever you want. So that's, once you go to the table level for visualization, there are no constraints. So it's just, so you can in that sense. Yes, so someone asked, why do I have a jar here, but I applied. So I'm taking material that is from other workshop and readapting here. In this case, this is these remain here. So in honor of Michael, we are showing here DSIC2, but there is a jar, VUM, different modalities, so a lot of flexibility.
Yeah, I use EdgeR and VUM all the time, so use what you like. Just I wanted to mention one thing before we get to the hour, and people can ask questions if they like. Or one thing is, if you're just getting interested in this, and you're like, oh, there's a lot of stuff shown today, we have a Slack channel in the bioconductor community Slack called Tidiness in BioC. And if you just want to like get started, there's a lot of people there who are happy to answer questions. So if you're like, I want to try this out. I saw this workshop, it was interesting, but I don't know where to get started. Like, yeah, there's both material, but you can also just talk to us. And I find it really fun challenge to try to rephrase a problem and make it like nice, clean, tidy code. That's kind of what I've been doing with all my teaching material and analysis code, just slowly move it all into this tidy framework. Tidiness. I think I spelled it right. Yeah, like I gave Stefano a challenge a couple months ago or something, like earlier in the year. Something complicated with NEST and multi-omic single cell data, and he found a solution. Oh, and bioconductor, yeah, just that's a good question. So there's a community bioconductor Slack, and I bet if you Google it, like BioC community Slack, it's pretty much an open Slack channel. And there's a lot of developers there. There's a lot of users there. It's a very, open community. Probably one of the best things about bioconductors. People are excited to get, bring people in, get them using the code.
Yeah. Well, one thing it would be nice to mention for this crowd is that the development is continuing of these tools, and I for sure can speak for the tool I show you. So if someone is, and paper have to be published as well. For example, we are missing the, and maybe, this could be a common effort actually while we were discussing, but anyway, so the bioconductor tidy summarized experiment and single cell experiment adapters have not been published yet. But the bottom line is that we are looking for developers of new hands that might be familiar with, our programming and might want to join for whatever project, can become a PhD project or so on. Get in contact with us. That will be welcome.
Oh, there's a question from May. Maybe Stefano, can you unmute? I think
Yeah, okay.
you can do that.
Yeah, yes. Well, hello.
If you right click or something, there should be an ask to unmute.
Ask to unmute. Yeah, sorry. Yeah, I clicked.
Hey, sorry, I just wrote it in the chat. I didn't realize that I could do that. I just wondered, can you import 10x Genomics VDJ sequencing into the tidy ecosystem? Hello?
Yeah, sorry.
Yeah, we can hear you.
Yes. VDJ sequencing tidy transcriptomics. Well, the short answer is whatever this single cell container can include, this tidy representation can represent. Okay, so are just a layer on top. So for sure, VDJ in single cell experiment is treated well. In Surat is a bit more straightforward. Actually, in single cell experiment, I have to say that the tidy representation still doesn't show the it's called alternative experiment. This will change soon. Actually, this became an interesting question because some development is needed. For sure, for Surat is just another assay and is a bit more modular in that. Yeah, for single cell, I will take care of the alternative experiments where these data might be hosted. But if this VDJ is hosted in the main assay in single cell experiment, this will be treated as you know, a standard one.
There's a question, a bootstrapping question. So Peyton asks, what step do we begin the bootstrapping? For example, what if the scores themselves are from a model? So I think actually it's really non-trivial to think about what aspect of variability you want to pass to the statistic that you calculate. And so we often find ourselves, like Eric and Wenson and Doug and I, thinking like, well, what is this? Is this like a matching problem or a bootstrapping problem? And like, as you come up with as well. So I think, you could, for example, like not bootstrap the features with the block bootstrap, but just, do like multiple imputation of the scores alone, maybe. Or maybe you also want to have some mixing of the locations. So I think that aspect of specifying the right null model for your problem, it actually just takes a lot of thinking and like talking to other people. What do you think is important? Or maybe it's important to like be controlling for some confounding variables. And so I don't have a very good like set answer for that, but I think it's like, what aspect of the variability do you want to pass through to the null model? Oh, and then there's a question about spatial omics.
Yeah. Also, Valeria has raised hands, but I will answer the spatial omics. So yes, that one of the further developments we might push if we have, I personally have more hands with us, but well, spatial, for example, if we are talking about bioconductor, the spatial experiment is, I think, a spin-off of single cell experiment. So it will be represented. Now, I haven't tested, so it's compatible. It depends on if you use, other internally, if you use some other organization. So I don't know, but everything that is coming off summarized experiment and single cell experiment are compatible with tidy. For Surat, I believe, again, it's a bit more modular. So Surat can host spatial coordinates. Again, I don't want to answer improperly. So I would say this is for sure a future development and testing. How to make a summarized experiment a tidy summarized experiment. So in the initial presentation, which I will briefly share because I'm already on the, no, maybe I cannot share. But anyway, so I was mentioning that the question, I mean, this, well, we never go from a summarized experiment to a tidy summarized experiment. So we never touch summarized experiment. When we load the library, tidy summarized experiment, then this adapter will abstract the display and abstract the tidyverse functionality for us, okay? But your object will be always a summarized experiment untouched. And any package that work with summarized experiment will never know you are seeing a tidy version of that, okay? So it's just an interface for you. Basically just load tidy summarized experiment library and everything will be open then on that object.
Okay, I try to allow Valerie to ask.
Hello, I have a question to continue the discussion about VDJ data. Did I understand you correctly that you propose to treat this VDJ data in a more like sophisticated way than just add the most frequent T cell receptor or the most frequent B cell receptor in the metadata of the object, like kind of zeroed object, and then is it correct?
I mean, yes, you. I mean, what you just proposed, for sure it works. Okay, so you can add. It depends how you want to represent this data. If you have counts for single cell, this can be treated as genes. I have worked with VDJ data, T cell receptor, but not extensively. Okay, so my experience is quite limited here. But anything you add to the metadata will be treated as metadata, so cell-related information. If you want to add the counts, I think you can include those in a SRAT container and it will be treated and visualized as I just showed in the workshop. That probably most as I can say, because I don't have very, very extensive experience. One thing I can for sure say is that for new maybe uses that I haven't I haven't explored yet, you are very free to send an issue to the related package on this my GitHub. There are all the packages are there. Maybe commenting, if something doesn't work or doesn't apply and so on. So we can keep the development based on the community needs.
Any other questions? Great. Please reach out to us if you want to, because also one last thing to mention, these packages all have pretty nice documentation that's in package down websites. It's actually much easier to see a lot of the documentation if you go to like the package down, like the home pages on GitHub or right, Stefano? That's true, right? For you as well. Yeah. Okay. And then otherwise, I guess we'll see you online.
Thanks a lot to everybody.
Thanks everybody for coming. And I'll post these videos later.